Ladies and gentlemen, good evening. I'm Anne Greenoff and I'm Head of School of Medicine, and it's my pleasure and privilege to welcome you to this inaugural lecture. Inaugural lectures celebrate the highest academic achievements. This inaugural lecture will be given by Professor Rebecca Oakey. Professor Oakey completed her DPhil at Oxford University. She then left the United Kingdom for the United States, during which time she was a postdoctoral researcher at Duke University Medical Center in North Carolina, the University of Pennsylvania, and the Fox Chase Cancer Center in Philadelphia in 1996. Rebecca was appointed Assistant Professor of Pediatrics at the Children's Hospital of Philadelphia. We were delighted that in the end of 2001, Rebecca returned to the United Kingdom to take up a lecturer's post in the Department of Medical and Molecular Genetics at King's College London. Her research program aims to understand aspects of the basic principles underpinning epigenetic factors in cellular processes using model transcriptional systems and discovering how errors in epigenetic processes can contribute to human disease. I am delighted to invite Rebecca Oakey to give her inaugural lecture entitled Epigenetics, a Journey from Mechanisms in Biology to Human Disease. Professor Oakey.
Can you hear me? Yes. Well, I'd like to thank you all very much for making the journey here today, and thank you very much for a warm welcome. And I appreciate that some of you have come from a long way in my past, as well as some of my current colleagues, and I very much appreciate that. So I'm going to start, as Anne said, by discussing epigenetics, a journey from mechanisms in biology to human disease. And just by way of outline, I'm just going to tell you a little bit about the early years that formed my scientific thinking, some genetics and epigenetics definitions for those of you that don't know about them, a little bit of a historical view of epigenetics, which is a relatively recent area or field. I'm going to talk about some mechanisms that we work on and how ultimately they can be applied to the understanding of human genetic disease. So this was one of my early scientific influences. This is my mother in her lab. She always wore that lab coat. In fact, she used to pick me up from school wearing that lab coat. In her Mini, she was a little white form, late from the lab, picking me up. And actually, for a while, I thought everyone's mother wore lab coats, but it turns out they don't. So she was obviously a very central influence on choosing science as an option quite early on. So there were, when I went to Oxford, three, I guess, major influences again on me while I was doing my postgraduate training. And first and foremost is Chris Tyler-Smith. He was my supervisor. He taught me how to think. He taught me how to do experiments. And he had a profound effect on the way that I think about things today. And to him, I am always very grateful and indebted. And the head of our unit was Ed Southern, who was also an interesting man. He was very eccentric. He, at the time, in 1986, was actually in the lab with his lab coat on, sticking bits of glue and string onto microscope slides, pioneering the first microarrays, which were a good 10 or 15 years before they became something that we used routinely in the lab. And finally, there was this other guy who was reported in the national press as having hopes pinned on him by Oxford University in the form of some type of golf championship. And I have to say, over the past 25 years, I've pinned a lot of hopes on him, quite often, and he's usually managed to pull through. So this was where we began here in the Department of Biochemistry, which was the original Department of Biochemistry, which was built in the 1960s. It had very drafty windows and pale blue paint, and it was on South Parks Road in Oxford, which I'm sure several of you know that building. After finishing with Chris and Ed, I flew to this southern state in the United States, North Carolina, where I was at Duke University as a postdoc with Mike Selden, who taught me how to do mouse genetics. 2 or 3 years later, we took a little flight up here to Philadelphia. This is the Liberty Bell, the city of brotherly love. And as Anne said, I did 2 further postdocs with Bob Nussbaum, a human geneticist, and Murray Brilliant, who was, again, an expert in mouse pigmentation. This is the building where I had my lab. This is the Children's Hospital of Philadelphia. And in 2001, the end of 2001, we flew back and arrived at King's College London, and London was about as foreign as you can get, as far as I was concerned. I'd lived away for 13 years, and people behaved and spoke differently. I went to North Carolina with 2 suitcases, Pan Am, lost one, and I actually only had, everything I owned was in something about that size. But when we returned, this is a 45-foot shipping container, and in there was the contents of our home, 2 labs, 2 offices, the combined bits and bobs of students and postdocs who were coming with us, and amongst the furniture, there were some extra people. So why am I interested in genetics? And I think this representation by David McCandless that was recently exhibited at the Wellcome Trust does illustrate some of the reasons why genetics is very important in our society. This is a proportional representation of all the things that people die from, and it's actually proportional for the last 100 years. So if we look, we can see that birth defects, which we think of traditionally as being the genetic component of human health, occupies a sizable but perhaps quite small circle in this total series of pictures. But in fact, if we think about it, genetics contributes to many, many, many reasons that we die: cardiovascular disease, a whole slew of diseases that are non-communicable, cancer, and of course infectious disease, where host genetics plays a role in infection. And that doesn't even take into account all the individuals who are living with genetic disease but die from other things. So bearing that in mind, there was an international effort for many, many years, costing millions of dollars, to bring together the sequence of the human genome, which culminated in a draft in the year 2000 and a completed sequence in 2003. So now we had a complete understanding of the human genome. We know all the bases in a typical human person. Let's see how that affects human disease. But of course, it wasn't quite that simple. And over the next decade, culminating in a series of 30 publications in September of last year, there was a huge effort to try and understand the function of those sequences which were in the human genome. And this was called ENCODE. And I mention it now because I'm going to come back to it again later. So whilst we know the sequence of the human genome, what we don't know is really what those sequences can do. It's quite a timely moment for the King's College London's history, because last week marked the 60 years of a very important finding in genetic research, which was actually it's not the discovery of DNA, but the structure of DNA, which was in fact facilitated by this rather beautiful picture, this diffraction picture, which was generated by Rosalind Franklin. She was working at King's College London when she made this beautiful photograph, and it was in collaboration with her colleague Maurice Wilkins and with Francis Crick and James Watson that the structure of DNA was elucidated. And once the structure was elucidated, we had a much better understanding of how this DNA molecule then might work in heredity. It turns out that that wasn't the answer either, because now we had the structure, lots of chemistry went on, and even some biology with enzymes, all of which contributed to being able to translate or to understand what this helical structure was in terms of DNA sequence and the bases that make up the genetic code. And the conclusion from all of this work was that there are 4 bases, G, A, T, and C, and they form these lines and lines and lines of sequence, which we now have to try and understand how gives rise to life on Earth. So what I'm interested in are not these 3 bases, but this one, cytosine. Cytosine is the most important base in mammals, and the reason it's important in mammals is because it has the chemistry that allows a methylated group to be added to part of the molecule. And I'm interested in what this modification of DNA on the cytosine can do for development and disease. So here's the cytosine normally, and here is its extra methyl group, which sits right here. And this only happens in mammals, or usually happens in mammals, when the cytosine is sat next to a G. So in a CpG dinucleotide, we see the modification of the cytosine. It's just important to remember that the CpG is something I'm going to refer to later. So in the genome, the cytosine bases are usually methylated. They usually have one of these modifications, and they are present throughout the genome, except in little places that constitute up to about 1% of the genome that are called islands. And the reason they're called islands is because they are literally bumps in the sequence that exist in a big sea of methylated cytosines. These little bumps or islands are very important because they sit, and they're always green when you illustrate them, and they sit at the promoter of genes. That's the bit of the gene where the transcription starts. So they sit here, and they're unmethylated, and they permit genes to be expressed. As convention has it, we will represent this CpG island in this format, which I just want to explain. If you see lots of circles that are black, it means that the cytosines are methylated, and the convention is that means the gene is off. And the convention for unmethylated cytosines in big groups like this is the island is unmethylated and the gene is switched on. So as it turns out, epigenetics is all about DNA methylation, but it is also about something else. These are proteins. They're modifications of proteins which play an important role in orchestrating the function of a cell. So it turns out that you have 3 billion base pairs of DNA in your cell. If you put them all in a row, that makes 3 meters of DNA. And as you can imagine, cells are very small. So in order to cram 3 meters of DNA into every cell, it needs to be very, very, very carefully wound around a series of very regular and very important packing proteins. And these are called histones, and they have a very interesting feature in that they have these little things that poke out of them. And on the ends of these things, they have the ability to be modified. And again, it's like a modification that we just discussed. It changes the properties of these proteins. It changes their ability and their behavior in the cell. So what is epigenetic information for? What does it do? So first of all, we have to appreciate some of its properties. It's very stable in somatic cells. That means every cell in your body that divides has the same epigenetic marks. And if it doesn't do that, then there are certainly problems that are going to happen. These epigenetic marks are reprogrammed in germ cells and early embryos, which means every time that we reproduce, we reset this epigenetic program so that the new organism has a new set of marks. The usefulness of this epigenetic information is it controls, or it's involved with, pluripotency. So if we imagine this oocyte and sperm represents a fertilization event, Here we have some early cells that have just divided as a result of this fertilization. These cells contain exactly the same DNA sequence as each other. So one can imagine that they could divide forever, but they would look the same. But they don't, do they? They don't look the same. They divide and they differentiate into hearts and nervous systems and immune systems and all sorts of things. So in order to make a cell which has two cells which have identical DNA sequences into two different types of cell, ultimately through differentiation, you have to have some kind of mechanism for doing that. And the mechanism is to use epigenetic information to change the destiny of a developing cell. So those are the sort of main categories of main areas that people are interested in terms of epigenetic information. But there are some very interesting genetic model systems in which these studies have been used to elucidate the mechanisms and the properties of these functions today. So one of them is called genomic imprinting, which I'm going to talk about. One is exon activation, which I'm not going to talk about today, but is involved in balancing the X chromosomes in females, and transposon control. So genomic imprinting is an unusual phenomenon, and when it was first realized or discovered, it was discovered through the observation of people who look for deviations from Mendelian inheritance. So deviations from traits and coat colors and eye colors not behaving themselves when they're inherited. So two labs, one in Cambridge and one in Philadelphia, decided to explore this unusual Mendelian pattern of inheritance. And they decided to look at what happened when you compared an embryo that had been generated from an egg and a sperm with an embryo in mouse that had been generated from two oocytes, so for two maternal genomes, and an embryo that had been generated from two paternal genomes. Now, these can be achieved using similar techniques to in vitro fertilization. You fuse these pronuclei and end up with an embryo. This is a normal mid-gestation mouse embryo with its corresponding placenta. If you have two maternal genomes, you can see whilst an embryo can develop mostly the way to midterm, the placenta is insufficient to feed that embryo, and this embryo dies. Conversely, if you have two sperm genomes, the embryo is vastly retarded in size and development, but it does have this fairly substantial placenta. This is also seen in humans, where a parthenogenetic conceptus, two maternal genomes, form these, typically these ovarian teratomas. This is actually human teeth developing around a big knot of human hair. And this is something which was reported in a very scientific journal called Take a Break, which is usually seen in the grocery store exit. And in this case, the lady was told that she'd had a baby in her tummy for 13 years. So she'd actually had twins, and the teratoma was not detected, and she felt that she had been gestating for a very long time. You also see androgenetic conceptuses, which are called hydatidiform moles. This is a normal fetus here. This is a mole. The mole is consisting of these large placenta-derived cells which do not go on to form a baby. So these types of studies led to understanding that there's a functional non-equivalence of the parental genomes, which is it was linked in 1991 to the existence of individual genes that caused this functional non-equivalence. That is to say, while most genes are expressed from your mother's chromosome and from your father's chromosome, there are sets of genes which are only expressed from either your mother's chromosome, and they're silenced in the paternal chromosome, or they're expressed in the father's chromosome, but they're silenced in the mother's chromosome. And what these genes do is provide an excellent model. Studies over the years have shown that a large number of these genes function in the relationship between the placenta and the developing fetus in terms of nutrient flow. So they're very important in growth as well as development of the embryo and the embryonic tissues. So where we stood now in the history of this phenomenon is that it opened up the study of epigenetics. We know there are roughly in the region of 20,000 genes in the mammalian genome, but only 150 imprinted genes. These are essential for embryonic and fetal development. That's been shown many times. But what really mattered to people who were thinking about this as a model system is that it made us question the mechanisms of gene control because they weren't behaving in the same way as most of the genes in the genome. What made them an ideal model is that they have two identical sequences, because they're the same gene, with two different states. They're modified epigenetically in two different ways. One of these ways is to activate a gene, and one of these ways is to silence a gene. And this coincided with the birth of the field of epigenetics. So in the 1990s, there were tools that were used to study genomic imprinting, trying to force the situation where you could have two maternal chromosomes and two paternal chromosomes isolated away from the normal complement where you had one from each parent. And these are called uniparental disomies, things that cytogeneticists are very familiar with. And the way in the mouse genetics field that these particular types of special individuals are generated is with the use of something called a Robertsonian translocation chromosome. In the mouse, most chromosomes are acrocentric, so there's not any genetic material away from the centromere. So all the goodies are down here, the centromere is here, and this is an easy way for this funny chromosome to form. Because if you fuse these chromosomes together, they form this single chromosome, where this would be maybe chromosome 7, this would maybe chromosome 11, with a single centromere here. And forcing this to happen leads to something called non-disjunction to occur much more frequently. So it increases the frequency with which chromosomes do not know which way to go. A long time ago, back in the 1990s, there were these strains of mice that were purple and blue, carrying the same chromosomes, and then wild-type strains of mice in red and green, which could be intercrossed together to generate new strains which were no longer having a pair of these chromosomes, but had single chromosomes, along with some wild-type chromosomes that could mind their own business and choose which pole to go to during meiosis, whereas this poor chromosome was a bit kind of slow and didn't know which way to go, because this one wanted to go this way, one wanted to go that way. So if you do a mathematical prediction of what happens when you mix these up, you end up finding all sorts of problems, such as trisomies, monosomies, and combinations of both. And this happens in human populations at a very low frequency, but it can happen at quite high frequencies in the mouse, because these are naturally occurring strains of mice that run about, which people collect. So what we were interested in is this sort of balanced uniparental disomy for chromosome 7, so we could have our favorite chromosome, which had lots of imprinted genes, forced into a single genetic tool. Now, I was doing this at about the time of the old Bush was hanging out, so he was dictating what was going to happen in the United States. He obviously then had a son come into power, but this wasn't at that time. So for those of you who have done lab work for a long time, who are very old, you will know this is a laughably historic technique of finding out the parental origin of your genetic material. But it was laborious and painstaking, but it had a place in history. This is a way of finding out whose chromosome comes from where. It relied on running enormous, cumbersome gels with very, very long procedure times, and at the end of the day, somebody would then drop your gel and you'd have to start all over again. So throughout this big ordeal of genotyping for long, long time, I found a handful of individuals which had both chromosome 7s inherited from the mother, and a handful that had both chromosome 7s inherited from the father. And we used these to examine gene expression. I was doing this while this impeachable president was in the White House. So now we had some genetic tools to use, and genetic tools are very useful because very often they provide a way of looking at something in a novel way that you couldn't do before, or in an organism you couldn't do before. And as I mentioned before, Ed was busy in the 1980s trying to figure out how to make these microarrays, and by the time I'd come to the United Kingdom, people were using these every day in all sorts of labs. These were little tools which allowed you to look for gene expression from anything you wanted to, really. In this case, we used these uniparental disomies, for which we were looking at the expression from maternal chromosomes and paternal chromosomes in isolation. And if you look at the graph, you can see a normal gene would have a contribution from the mother and a contribution from the father. However, if you have a uniparental disomy, a given gene is only going to be expressed in this individual if it's maternally expressed, and any paternally expressed genes won't be there at all. And conversely, in the paternal uniparental disomy, only paternally expressed genes will be present. There won't be any maternally only expressed genes. And this was, by the time I came here, this man was around. So this led to the identification of an imprinted gene. Doesn't really matter what it is, except that we know that it was oddly located in the genome in an intron of another gene, which seemed a bit strange, but there you go. It was expressed in the brain. This is a section of a brain showing the expression of this gene here. The assay that we did, a molecular assay to detect the expression of this gene, showed that it was paternally expressed. And we were able to validate that with the assay that I showed you at the beginning, showing that in oocytes, or maternally derived cells, you get DNA methylation on the cytosines. But in sperm, or any paternally derived material, we see that there's no methylation, or very little methylation. And this suggests that there's expression from the paternal chromosome. What was also very odd was that this gene itself, not only being located in an intron, seemed to have a funny structure. So genes have a bit at the beginning where they start, the promoter, they have some exons, they're nice, and they have these intervening sequences which are spliced out to make a messenger RNA, and this is the juicy bit that makes the gene product. Everything else is just cast aside for a moment, and then that goes on to make a protein. Except in this case, these messenger RNAs were stolen by the reverse transcriptase enzyme that's made by a repeat in the genome called a line 1 element, and it was reintegrated elsewhere in the genome. And this is a very interesting process called retrotransposition, which normally is discussed by people who are interested in evolution because it contributes to the evolution of genomes. It produces new copies of genes which weren't there before. In some cases, they become pseudogenes. Most of the time, they become pseudogenes because they are not very good for the individual, and they're lost over time. They accumulate mutations, they disappear, and you can see them as ghosts in the genome. But once in a while, they become neo-functionalized, which means they get a new function. And the reason they might get a new function is because if they were coming from the X chromosome and they were landing in an autosome, they might confer some advantage to fertility, for example. When the X chromosome is silenced during spermatogenesis, if you're on an autosome, you might be able to express your protein and do something to make that lineage have an advantage. And in fact, that's probably what happens. But nonetheless, all of that doesn't matter. What we now see is a gene that's intronless. It doesn't have any introns. And that was surprising. So we thought about this. In fact, Andrew Wood thought about this, and he's just appeared over there in the corner of the room. He thought about this as being rather extraordinary. He couldn't really rationalize why you would sit in an intronless gene would sit in the intron of another gene with this CPG island over its promoter that was differentially methylated. It was methylated in oocytes and unmethylated in sperm. It was expressed from the paternal allele, and it had a parent gene on the X chromosome, and it really bothered him that it had some friends in the genome that were just like this. There were 2 other genes that had these same principles. And because he likes puzzles, he decided to put all of those things together in a Venn diagram. He took all the genes that were intronless but sat in other people's introns and put them in this circle. He took all of the genes that had a CPG island over their promoter and put them in this circle, and he put all the X-derived ones in this circle. And in the middle, there was this one gene called MCTS2. Turns out that his theory was right. He had figured out that this was a pattern. And this was perhaps the first identification of an imprinted gene using only bioinformatics, only sequence-based predictions, no experiments at all. That's why it got done so quickly. So this retrogene, MCTS2, is probably an oncogene. It sits in the intron of a gene called H13, which is the histocompatibility gene. Oops. We did the molecular test that showed it was paternally expressed, it was imprinted, and it had this CPG island overlapping its promoter, just like the other imprinted retrogenes. They had these features in common which were very compelling. So, having thought about this gene some more, we thought, well, we can't look a gift horse in the mouth and have a little stab at trying to understand it. So the human genome is estimated, as I said before, to have about 20,000 genes. It produces about 10 times this number of transcripts. That means every gene has to make lots and lots of different versions of itself in order to fulfill the needs of a mammalian cell. But to do this, it has to use some tricks because it can't just be copying itself over and over again. So the tricks it uses are alternative promoters, which means it can use alternative things at the beginning of a gene to make lots of different transcripts. It can use alternative splicing, which makes different products from the middle of the gene. And it can use alternative polyadenylation, which makes different things from the end of the gene. And this gene we're going to talk about has a feature that uses the third option, which is alternative polyadenylation, to make all sorts of different versions of the same gene, because it ends the product, ends the transcript in different places. And these different transcripts can have different functions. So this particular host gene, the gene that MCTS2 sits in, made several transcripts. It made short transcripts, and it made long transcripts. And that was interesting, short and long, that's okay, we didn't mind that. But then when you look at what's going on, we could see that these are the polyadenylation signals, the 5 polyadenylation signals that made these short and long transcripts. So when we looked at it more closely, we could see that the short ones were all on this side of the retrogene and its promoter, and the long ones were all on this side of the retrogene and its promoter. So a closer look at this locus revealed, in fact, that DNA methylation was playing a role. That promoter was doing something to this gene. So all of the transcripts with their 3' end upstream of this CPG island were paternally expressed. And all of the transcripts that had their 3' end downstream of the CPG island are maternally expressed. So something is going on here with this promoter to affect the way these genes are being transcribed. And if you look at this model, you can see that this is just a cartoon of the previous slide where anything that's maternally derived uses this canonical polyadenylation signal, makes nice long transcripts, but anything that's paternally derived is truncated right here, right at the site of the promoter of the internal gene. And the difference between this side is that this side is methylated and this side is unmethylated and permits transcription from the retrogene. So we're now hinging all all the epigenetic information hinges on this DNA methylation here. And it turns out that that was a very close look at a single locus, a very close look at something which was a particular gene in depth. But what we've subsequently learned, given that the conventional view is that promoters are located in the 5' regions of genes, this is in the promoter, recent technologies have really improved the ability to detect CPG islands throughout the genome. And these have revealed that half of the CPG islands in the genome are not at promoters at all. In fact, where they are at intragenic regions, or some of them are intergenic, but they are sitting in the middle of gene bodies where they shouldn't be. So if half the CPG islands are sitting in gene bodies, they're interfering with normal gene expression. And these locations are not just a few, there are thousands of internal or extra promoters, which represents a significant force on the transcriptome. This is especially relevant for tissue-specific gene regulation, and this is how we think, going back to the idea of the epigenome playing a role in turning undifferentiated cells into differentiated cells, these internal promoters are playing a role in the differences between different tissues. And this just illustrates the utility of understanding mechanisms well on a single place in the genome before looking for applications to much more global function. Now, on the level of the locus, this very complicated slide, which has been designed by Mike Cowley, who has an interest in this locus, is to actually look in detail at what happens when you take away this internal promoter, what happens to the host gene, and if you move the promoter to an innocent gene somewhere else, what happens to it? So there are lots of longitudinal experiments to continue to drill down on this type of mechanism. And not forgetting that histones also play a role in epigenetic mechanisms, we must remember that these marks, doesn't matter which mark this is a mark that happens to be involved in elongation, and it's a modulator of splicing, one of the mechanisms that's linked to polyadenylation. These protein modifications can slow down the whole process of transcription, and so it's important then to manipulate these in a genetic way, and together these approaches improve our understanding of how intragenic promoters and epigenetics make multiple transcripts across so many different tissues. So that's thinking about the single locus, but what about across the genome and the epigenome? Again, we've had, yeah, it's nice, isn't it? So we've got the idea of the human genome, it's completed, we know all about it now because ENCODE's told us every functional aspect of the sequence of DNA. Well, of course, that's not true. We're beginning to chip away and understand some of the proteins that are involved in the epigenome, some of the DNA methylation patterns that are involved. We're trying to understand how they direct differentiation. But what really is next? So ENCODE, this is a cartoon from the ENCODE publications, and these are just some of the histones, showing the histones that we were talking about, and DNA methylation is represented on there somewhere. It's sort of looking for landmarks and functional domains in the genome to try and make sense of all that linear sequence. So because we can now do sequencing in a very different way to the sequencing we did 13 years ago to complete the human genome, high-throughput sequencing generates billions of base pairs, billions of sequenced genomes every day, all over the world. So you can have your genome sequenced, where you'd get nearly all the sequence of your genome. You could have your methylome sequenced, so you could find out what your methylation patterns look like. You could have your epigenome map, so you could find out what proteins bind to your bits of DNA. And what people are doing a lot of at the moment are exome sequencing only the juicy bits. They represent the bits that I've been talking about that get left in when you cut all the other bits out. This is a sort of high-throughput way of finding very rapidly disease-causing mutations. And then emerging is the looking at the transcriptome, just trying to find out what the RNA is that we are generating and that's different between us. And in our efforts to look at some of these, we're using DNA binding proteins, proteins that bind DNA, because we're quite interested in proteins that insulate between promoters, because that links back into our understanding of internal promoters. How do they get controlled? How are they stopped from misbehaving? How do you stop a promoter from misbehaving? And perhaps you could use a protein that insulates it, just like in lower organisms, such as yeast, where you can actually bind these important proteins and stop the crashing in of other promoters. And this work is being carried out across the genome, looking at lots of promoters to try and understand how these proteins interact, how they form differences between alleles, and try, therefore, to compare those different protein binding patterns across tissues so we can understand how stem cells differentiate into other tissues. So in particular, we've been studying the heart. There are some interesting cells in the heart which are called endocardial cells, and they differentiate. So this is the progenitor cell here. We go along this lineage, and at this point, a decision is made between these three types of cells. This decision to make the endocardium is really only based on one genetic marker. Otherwise, it would become endothelium, but we don't think that's enough to make this switch work. So actually what we're doing is sorting cells which look like this, and sorting cells which look like this, putting them into a tube and asking what's the difference between the DNA methylation between these two cell populations, what are the different histone marks, and we're matching that to its transcriptome and asking what makes these cells make this choice to go down the green cell lineage. And the reason that's important, or at least we feel it's important, is because the heart is continuously remodeled during development. These endocardial cells have the potential for repair and therefore are amenable to regenerative strategies. When we think about epigenetics in genetic disease, we know, for example, this disease here is caused by mutations or epi-mutations in a gene that controls methylation of a whole bunch of imprinted genes. We know that this disease here can be caused by either genetic or epigenetic mutations, and we don't know what this disease does at all, but at least it has there is some research that's going towards trying to understand if there's an epigenetic component, because these diseases have all got a component of impairment of growth, which we already know is associated with imprinted genes. There is some evidence that IVF has an effect on when cells are in plastic. They don't really like to keep their epigenome as neat as they should, and there could be effects on the epigenome for individuals who have IVF, who result from IVF. And also we know that there are many common complex disorders in which studies have been looked at. Studies have generated large amounts of data looking for regions which are likely to contribute to these diseases, and in many cases their favorite regions end up mapping to places where there are no genes. And so what we're interested in is how does epigenetics fit into this? How does it provide this missing heritability? So our goals in this area are to identify epigenetic differences between normal controls and individuals with disease, to match their transcriptomes to the epigenetic maps, to correlate DNA variants with these things, to understand the underlying molecular basis for disease, and to identify the missing heritability in these complex disorders. We'd like to be able to develop diagnostics, and epigenetic defects are potentially reversible because they have the nature of these marks is that you can reverse them because you can take them off during reprogramming of the germ cells. You should be able to take them off during cell division. And epigenetic factors in tissues that undergo remodeling are amenable to regenerative strategies, and that's something which is very plastic when it comes to epigenetic mechanisms. The reason that I found this it's been an amazing environment to work in is I've had many opportunities here. I've worked in this building that doesn't really sit in a Petri dish, but it is nonetheless something about this building that has enabled me to do what I do. There are a fantastic number of scientists in this building, amazing scientists doing all sorts of different things, which at first you don't think are very likely to be relevant, and after you spend some time with the people here, you realize that there are many avenues of collaboration. It has a fantastic patient resource. These are 3 of the major hospitals in London are allied to the same academic institution, and so we're very fortunate to have a fantastic access to patient samples. We're building a bioinformatics strategy that allows computer science to play a big role in the genome studies that we would like to continue. And the facilities are really excellent here. It's been very easy to do the things that I've wanted to do. And I think for the things that I'm interested in going forward, this post-genomic era, post-epigenomic era has certainly laid the way for the future, where we're having to think about genomic medicine and personalized medicine. Going to see your GP and having your genome given to you in a little piece of paper is something that's going to happen over the foreseeable future, and understanding how to deal with these large amounts of data and how to interpret them and how to think about how they contribute to stem cell differentiation, how they contribute to development and human disorders is a very important component of what happens here. So I have to thank my current and past lab members. We'll start here. This is, where is it? There's Mike Cowley. Mike Cowley is now a London Law Fellow. He's been a postdoc with me for the past few years. He's just been awarded this very prestigious fellowship, and he's primarily involved in all the work that we've talked about in imprinted retrogenes. This is Adam Prickett, who's actually, his work I haven't really talked about today in great detail, but he's worked on the genome analysis of insulator proteins, and he's now working on some human genetic disorders, and he's moving into his postdoctoral period now. And he told me today he'd been in the lab 4.5 years. It seems like only a couple of months, Adam. This is Nick Barkas. Nick straddles the fence, literally. So he is an experimental scientist and a bioinformatician. He's actually quite reluctant to come and solve your computer problems, but he can really analyze sequence data. This is Samuel, who is from Italy. He's an Erasmus student who's been a man with golden hands. He seems to be able to do everything faster than everybody else. So we are valuing him at the moment. This is Siobhan. Siobhan has been doing a PhD with me for the last 2.5 years. She is doing some high-throughput ChIP-seq analysis, which has finally paid off. Her hard work, she's nodding, it's paid off. This man doesn't appear to have any legs, but that's because he wasn't actually, he seems to be part of Siobhan's arm, but he was actually not in this photograph because he has joined us from another lab and is now doing some bioinformatics and network analysis to complete his PhD. This is Rachel. She was one of these. She's a BMS student. She just finished her 3-month practical with us. And who else is on the list? And I have to thank all of the intercalated BMS summer and work study students over the last 10 years, because there's been hundreds. And Andrew Wood, who actually noticed with Rowley Roberts that there was a funny thing going on with this gene, didn't have any introns. So he really initiated all this work while he was a PhD student with me. He's gone on to do many wonderful things. He's been a Sir Henry Wellcome Fellow and is now in Edinburgh. Ruth McCall, who did her PhD with me, she is now at Harvard, and she was part of the Insulator project. Trevelyan Menheniot, who went to Australia, but he was a gene cloner. Catherine Woodfine, who did all of the microarray experiments in the days when other people couldn't do them, and many other past members of the lab who we, of course, miss. This man, he didn't like this picture for some reason. This man he came to the UK from the University of Maryland Computer Science Department, as a postdoc and has stayed on. He's now a senior lecturer in the department. He is a fantastic bioinformatician. He's a bioinformatician who understands the biology. He was forced to do a year of lab work, and I think that's really paid off. So Rainer is an amazing collaborator and a very good friend. I need to thank all of our collaborators, of which there are many, from lots of different institutions who along the way have participated in lots of projects and lots of interesting studies, from plants to humans. I want to thank very particularly all my colleagues in the I always forget the name of the division, it's always the department I think of, in the division of genetics and molecular medicine, because everyone has played a role in the way that I've been able to work and coexist, to our division chief, Jill Bates, to everyone who has made this a really, really fantastic environment for me to work in, and I've really enjoyed myself. I have to thank all the funders, because lots of people have given us lots of money, and that's always very handy to do the experiments. I'd like to thank my mentors over the years. These individuals have played a big role. I'd like to thank Marianne, Donna, Marisa, Ellen, and Bev. And then I have to thank everybody else who's also played a big role in my time here. Lots of people who've been my friends, who've made me laugh, who've looked after my children, who've done things which are really beyond the call, and I can never thank people enough. And to my family, thank you very much.
I now call upon Professor Gillian Bates, head of the division, to give the vote of thanks.
Thank you, Anne. So can I invite you all to join me in thanking Rebecca for this really fascinating lecture. Rebecca, you have a true gift for making something that's such a biologically complex mechanism into something that we can all understand and appreciate and make it accessible to all of us. So you've taken us through this history of epigenetics and the many insights that you and your group have contributed to that field along the way. Perhaps it's a metaphor for competitive collaboration and how efficient that might be. That's the mechanism, not not you. So many, many congratulations on your personal chair, and thank you again for this very, very stimulating and entertaining lecture.
I think, ladies and gentlemen, you'd agree we've had a superb inaugural lecture. So please, let's go and celebrate in the foyer with the reception. Thank you very much.